over the macromolecules of proteins. So proteins account for 50% of the dry mass of a cell. So if the body was dehydrated, 50% of the body's mass would be proteins. So if you've ever seen a mummy, or if you've seen pictures of the bog people of Northern Europe, these people were thrown into a bog or died in a bog, and their bodies were dehydrated. So what's left behind are the proteins, which is about 50% of your body mass. Proteins are essential. We've got to have them. So they're going to provide structural support, they're going to be storage, they're going to help in transport. So if I have to move materials through a plasma membrane, remember these are all lipids. We talked about those in the last video. These will not going to allow water to go through easily, so I have to create enzymes that allow materials to go back and forth. It's going to help communicate with other cells. I'm going to be able to move information. If a cell needs to go somewhere or a cell needs to produce something, I'm going to be able to communicate. It's going to allow with movement. It's going to allow defense with foreign substances. So I'm going to be able to use proteins to be able to work with my immune system. I can create most enzymes with proteins. Those are going to go in and they're going to break down molecules. They can put together molecules. Enzymes really important for a lot of the stuff that our body does. The entire function of DNA is to make proteins. So DNA codes for proteins. So the genes code for proteins, and those proteins will make all those other things: storage, transport, work with our immune system. Proteins, very important for our body. We've got to have them, and we recognize when our proteins aren't there, we don't feel good, things aren't working like they should. Here I have a DNA strand, and here's another DNA strand, and I'm making these amino acids. Amino acids are the monomer of a protein. So these amino acids are going to come together, and they're going to make one long protein. If I have a change in my DNA code, if I change my DNA code, I'm going to change one of these monomers of protein. I'm going to change one of these amino acids. And if I change one of these amino acids, I'm going to have a different type of protein. And that different type of protein is going to fold differently. It's going to have a different function. And that's the big takeaway with proteins. If I change the shape, I change the function. And all of those are coded by my DNA. All of these codes are going to make individual amino acids, and those amino acids are going to come together to make one long protein. I have DNA. DNA is going to transcribe with RNA. RNA is going to go and it's going to make proteins for the body to be able to use. So again, DNA codes for proteins. When I have an amino acid, I'm going to have a setup structure just like this. Okay, I'm going to have an amino group. I'm going to have a carboxyl group. I'm going to have my R group. This is going to be the molecule that can be attached to my carbon. This can change. This part here will always be the same. I'm going to have one hydrogen atom, and I'm going to have an alpha carbon right here. Amino groups are basic, so why is it called an amino acid? So this is a basic group, but remember carboxyls, I have these oxygens, very electronegative. This hydrogen's here, it's going to pull off that electron, and this hydrogen proton is going to pop off and go into the solution, so this can make it into an acid. So that's why it's called an amino acid. And if you don't know pH, I have a video on pH that talks about that. There's 20 common amino acids, and they're all going to have different characteristics. I've got ones that are nonpolar. Remember, nonpolar means hydrophobic. I'm not going to want to be with water. It's water-fearing. I'm going to have polar, which is hydrophilic. It's going to be friendly with water. It's going to want to be around water. I'm going to have water that's going to have a positive charge, which means I'm bringing in, you see all these amino groups that I've got on here. These amino groups are going to bring in some of these, these amino groups down here are going to bring in hydrogen protons from the solution. So it's going to make this more of a basic. This one here, all these oxygens, see those negative marks right there? There were hydrogens on there, they've popped off. They've gone into the solution, so I'm going to have more of an acidic solution, and my molecule is going to be charged. So what do they all have in common? you notice they all have the very same backbone. They're all going to be the same, except for their R group. These colored groups right here, these are their R groups. So everything will be the same except for those R groups. I have nonpolar. These are not going to want to mix with water. So when these amino acids link, one's going to hook with the other. And then these side chain interaction, these R groups, are going to react with each other based on their properties. Nonpolar, remember, like likes like. So nonpolar is going to want to be with nonpolar. Polar is going to want to be with polar. So these polars are going to attract to each other. These nonpolars are going to be attracted to each other. These charged, I have negative, I have positive. Opposites attract. This negative charge is going to be attracted to this positive. Positive and positive are going to repel. Negative and negative are going to repel. So it's going to fold in a very specific way. So those are all the same except for those R groups. They're going to bond with dehydration synthesis. Remember, we have dehydration synthesis. Water comes out. So here I have an amino acid. Here I have another amino acid. This hydrogen from this amino group is going to pull a hydroxyl off the carboxyl side of this molecule. Remember, carboxyls, and we have amino groups. So these are going to pull off, and I'm going to have water taken out. This is going to produce a bond between the nitrogen and the carbon, and that's called a peptide bond. So this peptide bond is going to be due to dehydration synthesis. Amino acids are going to join the amino to carboxylin. So this carboxyl side is going to attach to this amino side, carboxyl to the amino, carboxyl to the amino. So it's going to keep going that way. My R groups are going to be off on the other side. I'm going to have two ends. I'm going to have what's called an N terminus, and I'm going to have a C terminus. So all amino acids, when I have a long polypeptide chain, one side is going to have my amino group, and one side is going to have a carboxyl group. So this is a C terminus, this is an N terminus. Polypeptides are going to bond those two together. Now if I wanted to break these bonds, what do I need to introduce to the system? I'm going to bring in water. And if I bring in water, then I'm going to put that hydroxyl on one, hydrogen on the other, and then I'll break this bond down. So every protein will have an amino group, an N terminus side, and a free carboxyl group, a C terminus side. There's 4 levels of proteins. You have what's called a primary structure, and you need to think of this as just a big long chain of pearls. All of these little pearls, individual pearls, are amino acids. So here I can see these are all amino acids, and they have to be put in a very specific order. And when they're put in this specific order, those side chains are going to interact. So that's going to pull in those beads. They're going to fold and change and convert into a very specific structure. And that very specific structure will have a very specific function. Again, I change the structure, I'm going to change the function. Secondary structure is going to be between hydrogen bonds and polypeptide backbones. Here I'm going to have beta pleated sheets, and these are kind of going to be like accordion hooked together. I'm going to have alpha helixes. These are going to curl and kind of go into like a little curly cube, but I'm not being set up by my R groups or my side chain interactions. These are going to be between the polypeptide backbone. That's my amino, my carbon, my hydrogen, my carboxyl group. Those are going to interact. So here, my carboxyl, my amino group are going to create a hydrogen bond. Remember, those dots represent hydrogen bonds. And that means this has a negative charge, this has a positive charge, and like magnets, they're going to get pulled towards each other. So I'm going to have them happen with beta sheets. So that's kind of like an accordion. They're going to fold in on each other. I'm going to have the same thing happen with alpha helices. They're going to curl. And as they curl, these hydrogens are going to hydrogen bond with those oxygens in those curls. Tertiary structure, this is going to be where my R groups, my side chains, are going to interact based on their properties. So it's going to be three-dimensional. It's going to be in a very specific shape. I can have ionic bonds, These are going to be ones where I have a positive charge and a negative charge, and they're going to attract. Remember, opposites attract. I'm going to have hydrophobic interactions. These are going to be all my hydrophobic molecules. They're all going to come in together, and you see how this kind of makes a pocket? And this pocket is going to come in and it's going to pinch off, and it's going to keep water out of this area right here. I have ones right here where I'm going to be polar. They're going to allow water to come in. I'm going to create a hydrogen bond between those. And here I have disulfide linkages. Disulfides, I have a SH, SH. Those hydrogens leave, and then I get a true covalent bond between those two sulfurs. That's a pretty good bond. So the fold, very specific, beta sheets, alpha helices, all of those are going to be due to the hydrogen bonds between these backbones. But my tertiary are these side chain interactions that are going to start pulling these beta sheets to an alpha helices, or this alpha helices towards this one. These side chain interactions are now going to start pulling this big long chain into a specific fold. So again, shape determines function. That's the big takeaway with proteins. The fourth level is quaternary, 2 or more tertiary in one protein. So here I have a tertiary, 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 tertiary. So I have my primary structure, very specific set of amino acids. I'm going to have my secondary. I'm going to have beta sheets. Alpha helices are going to start working on those backbone hydrogen bonds. And then my tertiary, my side chain interactions are going to start to fold this in a very specific way. Most proteins are going to stop at tertiary, but there are a few that will go to a quaternary level. Hemoglobin's an example. So hemoglobin, I'm going to have 4 different tertiary structures that are going to come in, and they're going to bond together. So this is going to have a very specific function for hemoglobin. If I'm going to break down a protein, I can break down the protein with heat. I can change my pH and break it down. So a normal protein can be like this. I'm going to increase my temperature. I increase my temperature, I'm breaking these bonds down, and they won't go back together. So once it cools down, it's done. I can change my pH. My pH will change the charge of some of these bonds, and it's going to start pulling these bonds apart. So when you denature something, you can't renature it. It's done. It's like taking an egg, and I take an egg and I put it on a hot skillet. That hot temperature is breaking those bonds, is messing it back up. I can't get that egg to go back to its original state. I have denatured it. So your body can do that. When you break down proteins, you can change that. So if your pH goes off in your body, if you are sick and have a temperature, the change in temperature, that change in pH can change the proteins you have, your enzymes, how they work, and then you can have some medical issues due to that. Okay, that is a breakdown of proteins. If you guys have any questions, let me know. Talk to you later.